cognitive patterns, there are well-replicated findings on impairments in theory of mind, joint attention, and to a lesser extent on central coherence and executive planning. And we have had papers during this meeting which indicate the progress that has come from a greater use of experimental designs, the application of eye tracking methodology, the use of functional brain imaging, and the baby sibling prospective studies. At one time, the hope of many people was that it would be possible to identify a singular, modular cognitive deficit that fully accounted for autism. But I think it is now clear that that's rather unlikely to be the case. Kathy Lord, in her excellent talk this morning on DSM-5, talked about the subclassification. The concept of Asperger's syndrome was, in my view, useful in indicating that autism can and does develop in individuals who gain speech at a normal age. Nevertheless, it has to be said that the subclassification of autism spectrum disorders has proved problematic and lacking in adequate empirical validation. The DSM-5 working parties have argued that it's time to dispense with all the subclassification and have just one diagnosis of an autism spectrum disorder. Well, there is much to be said for this simplification, but there will be a need to have some way of identifying disintegrative disorders, not because we are sure that they're different, but rather because if it doesn't appear anywhere in the classification, it will block research determining whether this group is simply an unusual variant of autism or is something quite different. And the literature on disintegrative disorders is really quite small and inconclusive. Quasi-autism was mentioned by Eric Fonbon, for example, and the UK study of English and Romanian adoptees showed, much I have to say to my surprise, that profound institutional deprivation was associated with a clinical picture that was similar to autism, but atypical in some features. The mechanisms are not well understood, but the implication is that autism may develop on the basis of externally imposed restriction of stimuli, as well as an internal genetically influenced impairment in the processing of stimuli. It remains to be determined whether abuse and neglect in the family can have the same meaning, and comparative studies between deprivation of a severe kind in institutions and comparable degrees of deprivation in family settings is much needed and is yet to be undertaken. The limited available evidence suggests that there are differences, but that's really a very tentative conclusion. In passing, I should just emphasize that although this finding is, I think, very solid and I think very meaningful, it does not mean that most cases of autism derive on the basis of institutional deprivation, because we know from hundreds of studies of autism that such a background is actually quite rare. We've had some discussion of the use of medication, and the point that I want to emphasize here is that numerous studies have shown that autism stands out from almost all other psychiatric disorders in showing no marked benefits of psychotropic medication on core symptoms such as impaired social reciprocity and social communication. Why? One possible implication is that the basic deficit does not involve neurotransmitters. We need to remember that the brain is part of the body, and that we shouldn't automatically assume that the risk mechanisms are confined to the brain. So the issue discussed yesterday in terms of possible immunological features would be an example of something that goes outside the brain, although it has implications for brain functioning. So what is involved? It's clear that autism is some kind of broader system disorder, so why on earth doesn't it work? Well, of course, it may be that we've used the wrong medications, and of course it is true that the medications are of some limited value for associated problems. But the enigma is why that seems to be all. Now, I want to turn from clinical issues now to genetic findings, and we have heard a bit about those during this meeting. There are multiple replicated findings that autism is associated with rare pathogenic gene mutations such as neurolichens, neurexin, and SHANK3. They account for a tiny proportion of cases, about 1%, and it has been claimed that nevertheless they are true causes of autism. The dilemma is that although the clinical picture associated with these genes includes autistic features, intellectual disability often dominates. And it is interesting that the findings have been written up in terms of autism rather than intellectual disability, although it could well have been the other way around. Now, of course, genes do not code for specific psychiatric categories, and pleiotropic effects are to be expected. That's to say, the old notion that genes simply had one effect, we now know from numerous studies, is not the case. Most genes have multiple effects, and therefore finding a lack of specificity is not in itself surprising. But the lack of specificity raises doubts on the extent to which the findings are informative with respect to most cases of autism spectrum disorders. Now, of course, there are examples where the study of rare single gene causes of a condition are informative more broadly. I mean, that would be true in relation to Alzheimer's disease, for example. Whether that is true in relation to autism, we don't know. Copy number variations. Eric Fombel mentioned these. Tiny, submicroscopic chromosome deletions or duplications known as copy number variations. Several studies have shown that CNVs, especially those involving deletions, are found in some 5 to 10% of cases of autism, and that rate is significantly higher than that in controls. The evidence indicates a causal role for CNVs in both autism and schizophrenia, and now in a paper published last week in ADHD as well. But important queries remain. Most CNVs arise de novo and therefore cannot account for familiality. And in relation to autism, we know that it is familial. That's to say, it runs in families, and it's not just that there is a genetic component. So it doesn't provide an explanation for that. Also, when inherited, the CNVs may be present in family members who are not affected by autism, so the causal effect is not necessarily determinative. But also, we have to ask what causes the raised frequency of CNVs. One has the same query with chromosome anomalies, which are also increased in autism, and with minor congenital anomalies where the same applies. One possibility, touched on yesterday, is that of raised parental age, but adequate studies to test that possibility have yet to be undertaken. So there's no doubt that the discovery of CNVs was an exciting discovery. I remain uncertain as to whether they will help as much as some have claimed in understanding the biological pathways leading to autism. But they might, and I certainly think that geneticists are mistaken in not paying equal attention to where these come from. It's not that developmental perturbations have to have a specific cause. They don't. But we need to ask, nevertheless, why they are arising in a particularly frequent number. The flavour of the moment in genetics, at least for some, is genome-wide association studies. This arises because it is now possible through technological advances to move beyond looking at candidate genes to looking at all the genes in the genome in order to detect susceptibility genes. Now, GWAS, as they're colloquially called, require huge samples, and inevitably they will give rise to many false positives. The big advantage of GWAS is that they have the possibility to detect novel genetic associations, i.e. not simply the candidates that we know about for one reason or another. But I have to say, as Eric Fombaugh commented, that the findings have been unimpressive so far in the field of multifactorial mental disorders. Moreover, because the identified susceptibility genes have been found to have very weak effects, it remains uncertain whether the findings will be very informative on biological causal pathways. So that Ken Kendler, for example, talking not about autism particularly, but of all of these findings, comments that the odds ratio has generally been of the order of 1.1, 1.2, the most 1.3. So to talk about these as genes for schizophrenia or autism or ADHD is really going way beyond what is justified, relevant to each of these disorders, but only as part of a broader picture. Epigenetics is the other newcomer on the block, and there is great interest in the possibility that many genetic effects derive from epigenetics rather than from changes in gene sequence. Epigenetics refers to neurochemical changes that influence gene expression, and genes can only have effects if the genes are expressed. It's both tissue-specific and developmental phase-specific. It involves multiple DNA elements, which for reasons that only geneticists can explain, they don't like to call genes, but they are DNA, so to my mind, why not call them genes? It also involves chance effects and environmental influences. There is limited evidence that epigenetic mechanisms might be involved in autism, but so far their role remains uncertain. Nevertheless, what we do know is that epigenetics is for real, and it is certainly important to look at it carefully. The difficulty in human studies is the problem of tissue specificity. People have argued that because more genes are expressed in the brain than in any other body tissue, that it may be adequate to do the studies on lymphocytes, but that's still to be determined.